turn the floor over to Professor Sask to do the introduction.
Great, thank you. It's really my pleasure to introduce Professor Lara Gamble, who's a bioengineering research associate professor at the University of Washington, visiting us today remotely from Seattle. And she's been on faculty there since 2004. She's a co-director of the NIH-NIBIB funded National ESCA and Surface Analysis Center for Biomedical Problems, so NESAC Bio. She's also the director of the Molecular Analysis Facility at the University of Washington. Her scientific research interests include surface modification and characterization, biomaterials, biosensors, and molecular imaging in 2D and 3D, which I'm sure she's going to share a lot about today. Lara Gamble has over 30 publications and has given many invited talks and tutorials on surface analysis. And I've been to some of these workshops at the Society for Biomaterials meetings, and I'm looking forward to hearing more and having her here to share with all of us. Dr. Gamble received her bachelor's in chemistry at UC Santa Cruz and her PhD in physical chemistry from the University of Washington. She did postdoctoral research at NESAC Bio and then took a position at Xiomix, a biotech startup in Fremont, California. Her time in industry was followed by a slight change in direction to work at the Space Dynamics Laboratory in Logan, Utah. And then eventually she returned to NESAC Bio at the University of Washington. So we're so pleased to have you join us here. And thank you so much. We really look forward to hearing about your research.
Great, thank you. Thank you for inviting me, and I'm excited to give a presentation. It's kind of fun to be able to do all of these talks without actually leaving Seattle. So I'm happy to give a presentation and talk with everybody. Yeah, I've had, oops, sorry. As was mentioned, I've had quite the interesting career, and I got to go backwards. I did initially start at NESAC Bio, and then I went out to work in industry for a while. And as was mentioned, I worked in Space Dynamics Lab. So I actually made something that went up on International Space Station. So that was quite a different turn of events. But I'd like to give you guys a little bit of background on what we do at NESAC Bio, what our motivation is, and also some of the current types of research that I'm working on right now. And that's why I titled it Getting to Know the Surface of Biomaterials. NESAC Bio is a long-running NIH-funded center that actually was, the main focus of the center is to take advanced surface analysis, surface science techniques, and develop them and modify them to be able to address biomedical problems. And so I'll be talking about a lot of that sort of thing that we do. But to take a step back a little bit, traditional surface science, if anybody isn't familiar with that, has its roots a little bit more in things like catalysis and the semiconductor industries. And for example, this is some old work that I did on titanium dioxide, and this was actually titanium dioxide often used for solar cells and a variety of other applications, and working on putting a coating on that, but understanding it down at the very basic level of where do bonds go, where do molecules go, is what we do in surface science. Or something as simple as taking what they call a silane coupling agent, which is a silicon with some sort of leaving groups, and how that will react with hydroxyl groups and water on a silicon wafer to form an organic layer with some organic functional group as a way to bond organic to inorganic substrates. And people do this a lot when they're talking about, for example, biosensors. So one of the biggest examples of that would be a DNA chip. These, if anybody's familiar with the DNA microarrays that are out there, for example, Affymetrix is one of the better known ones. They put down sequences of DNA on silicon wafer in little spots, and this could also be a protein chip, but in the DNA version, it's half of the DNA is down, and then they look for whatever the match is, and then they get a signal and they say, ah, that's the sequence of the DNA that I'm looking for. And so as a major part of that step, they have to bond DNA to an inorganic substrate, generally to a silicon wafer is what they do in Affymetrix chip. So that a lot of my initial work was done on working on that kind of a mobilization chemistry. I did this actually in industry, and then I brought it back to academia when I came to academia, where you'll have some sort of a substrate bond, some sort of functional group. It could be a silane system. It could be thiol gold type system, and then eventually make some sort of a functional groups on the surface that then can be bound to something that was on the DNA, and somehow you get the DNA spotted on the surface. So when people do this in industry, they actually want to be able to capture all the different sequences. So all these are little spots of DNA with different sequences, and the spots are about 100, 150 microns in diameter. And when you get a strong signal, you say, ah, there's a match, there's my DNA. And if you don't get a signal, you can say, oh, I don't have that sequence. But what does it mean when you get a ring or you get partial signal? And so what we're working on is how we can improve the reproducibility of this, and perhaps limit, pre-purification that has to be done so that you lose some of your precious DNA, and to better understand how that bonding system happens. And we did that through a variety of surface analysis techniques. And so I'm using this kind of older set of experiments to sort of introduce the techniques that I'm going to go into a lot more detail with some of our newer work. But the two main surface analysis techniques that we generally use are XPS and TOF-SIMS. So that's X-ray photoelectron spectroscopy, where you have X-rays in and they emit photoelectrons. Those electrons come out and we measure the kinetic energy, and that tells us something about what elements are present on the sample. And we can even quantify the percentage of those elements that are into the sample in the top 50 to say 100 angstroms of that sample. And you can also say something about, for example, molecular bonding environments. Is it carbon bound to hydrogen? Is it carbon bound to oxygen? You can get that from your XPS analysis. TOF-SIMS is a slightly different technique. We hit the surface with ions. So these are just ionized atoms or groups of atoms that hit the surface, and that often is referred to as the billiard ball effect, where you hit the balls that is the surface with a ball that's coming in pretty fast, and it knocks the balls from the surface off into the vacuum, and we measure the molecules that come up. So that's time of flight secondary ion mass spectrometry. And this gives a little slightly more surface sensitivity of it's reported as top 10 to 20 angstroms of a surface. So the XPS, which I just noticed it says spectrometry instead of spectroscopy, which is embarrassing. But XPS generally works by having X-rays in and electrons out, and then we will measure the binding energy of those electrons, and that tells us by different peaks what sort of elements that we have on our sample. So an example of that is what we did for the DNA microarrays. We didn't just take the spectroscopy. Well, we did take the spectroscopy where we have the different elements showing up in the spectra, but we also took an image of what was there. So we focus on a specific kinetic energy of electrons and map out where we see high intensity of those versus low intensity of those. And an easiest example to see here, this is an 800 by 800 micron square. We can see the little 100, 150 micron DNA spots, and there's less signal from the silicon where we see the DNA spot. In contrast, we see more signal from things like nitrogen and phosphorus, of course, being the backbone of DNA. And we see less silicon because we've attenuated the surface signal. So we can use this to map out where we see our DNA spots, but we can also use something like XPS, which is very quantitative, to actually quantitate how much DNA we have in those spots without putting in a fluorescent probe. And it can be very quantitative what we're getting. So we have two different rows here. This is DNA spots. You can barely see them, where there's no hybridization. So there's only half the DNA. Whereas in this one, we have a row where there's an unmatched. So there's only half the DNA again. But in this one, the DNA was matched. And so we see twice as much DNA in these particular rows. And you can see that by an increase in the signal that we're seeing in the phosphorus signal. And if you quantitate it a little bit more using some analysis, you can actually see that we've doubled the amount of phosphate that we have in those probes. So XPS can be very helpful in that it's extremely quantitative, and it's also potentially something that you can do in an imaging format. But if you want to know really a lot about imaging and good resolution imaging, we often go to time of flight secondary ion mass spectrometry. So TOF-SIMS, as I said, is an ion hitting the surface. It knocks molecules off the surface. This is kind of what a TOF-SIMS, this is our ion TOF-SIMS instrument. But how it functions is you'll have a gun that's producing ions. It's going to put a pulse of those ions that hit the surface. Then it knocks off molecules that go through the time of flight mass spectrometer, where of course then the smaller ones fly faster. And then we get a mass spectrum out at the end. And it can do a variety of different things are occurring there. We damage the surface a bit by doing that process. We get atoms, things break apart into their atomic structure very close to that impact point. But further away from that impact point, we can potentially get molecular ions. For example, the entire self-similar monolayer molecule, for example, might come off intact, and we can use TOF-SIMS to identify actual things that are on the surface, not just the little atoms that it breaks apart. So it gives us a variety of things. So for example, if you had a molecule A, B, C, D, near the impact crater, you might just see the components of that molecule. But as you go further and further out from where you had that primary ion impact, you'll see molecular fragments or potentially the entire molecule coming off intact so that we can identify it. So TOF-SIMS generally for biologics traditionally has produced a breaks down your proteins, for example, into the variety of different amino acids, just to be blunt. And for example, these are a variety of different proteins. We've got BSA, we've got collagen. They basically, these are spectra, TOF-SIMS spectra from different proteins. And you can see that they appear to have very similar peaks at similar masses. And the only thing that's different really is the relative ratios. So for much of TOF-SIMS, it can be difficult to identify what particular things are if you're not pulling out the molecular ion in some cases. So what we do to analyze a lot of our TOF-SIMS spectra is multivariate analysis of the spectra itself. So with our spectra, we'll get 50 to 500 peaks within one particular spectra. And to get a relationship between how those spectra change from sample to sample, we do a multivariate analysis, usually principal components analysis, but there's a variety of other ones that we use. And if you're not familiar with multivariate analysis, basically what happens is you pick out key peaks that you're looking at, and you'll say, what is the relationship between those samples using those key peaks? And you might find that they separate out. So this is sample A, separates out very well from sample B. There's actual line in between, and you can say, oh, there's different types of chemistry between sample A and sample B. It also tells you something about the reproducibility of the sample. So sample A, for example, all the different spots on the sample look very similar in principal component analysis. We have very narrow standard deviation saying that we have great reproducibility across that sample, whereas in sample B, there's a lot of scatter, indicating not as good reproducibility in that sample. And what we're looking at to understand what is separating out those samples is what we call the loads. And that shows the variables that are responsible for the differences. So these are the peaks that we were looking for. For example, if we were looking at cholesterol, if we're looking at different types of fatty acids, we can say, well, sample A is higher in, for example, cholesterol, whereas sample B tends to have more of particular types of fatty acids. And it tells us, informs us of how we can look for differences within the samples. We try not to use MBA as the final answer. However, using a type of multivariate analysis, in this case, is discriminant analysis, they were at, this is Matt Wagner, who's an old NESIC Bio student, was able to actually distinguish a variety of different types of proteins. And so you can use these types of techniques to really understand what is on your sample and differentiate between different types of biological samples. So why do we do TOF-SIMS for imaging? Imaging TOF-SIMS works where you focus that beam down into a pixel and then randomly raster it across the surface, mapping where you've hit onto the sample. And each pixel where you've collected data produces a mass spectrum. So imagine here, this is just a pile of cells on a substrate. Each pixel within this image is an individual mass spectrum, and I can pull out the mass of just cholesterol, for example, and find out where on this map we'll see higher intensity of that particular peak. Or I can do a comparative analysis and just say what parts of the image look different than other parts of the image. But the resolution that we can get with imaging TOF-SIMS is determined by how tightly we can focus this incoming ion source. And with some of the liquid metal ion sources that are out there, we can actually go sub-micron. And so we're getting, I think some of the newer systems are getting down to 100 nanometer resolution with that beam. So initially I did some of these, again, following with that DNA microarray example on some DNA microarrays. And you can see a few things. One is if we pick the peak for a particular ion, for example, phosphate PO2 or phosphate PO3, we can get a very good image of where we spotted that DNA. We could see ions for the individual bases of those DNA. And we can see some inhomogeneity within these images as well, where we might see a higher intensity of phosphate in one part and not the other, sort of indicating that the spotting process wasn't going along uniformly. Also, if you're looking at the non-complementary and the complementary within the same one, so this one has twice the amount of DNA than the other one, you can get some relative information from the relative intensity that this one seems to be brighter than this one. Therefore, we could say there is more phosphate here than this one. However, traditionally, it can be somewhat difficult to quantify with the TOF-SIMS analysis as one would for the XPS, and that's why we tend to pair the two together. All right, so using those techniques that we have, what do we do? What are we doing currently? And some of the work we've been doing currently has been related to nanoparticles. There's a lot being done with nanoparticles where people are making nanoparticles for a variety of different applications, drug delivery being one of the key ones that we look at because that's very much in the realm of the biomaterials and bioanalysis. And they put types of functional groups on the ends of the nanoparticles. In this case, this is the PLGA core of the nanoparticle with PEG on the outside. And in this case, they were putting on also surfactants within there. So they are not actually covalently bound, they were just surfactants. And they found that the surfactants, depending on what surfactant they used, they would be able to cross the blood-brain barrier or not. And some of them that surfactant will influence which of these nanoparticles can actually accumulate in the brain for being able to do drug delivery directly into the brain, which is a bit difficult. However, there's not much out there for how people can actually characterize their nanoparticles, the surface of their nanoparticles, and prove that they have that surfactant on their nanoparticle. And so we were working, this was actually Elizabeth Nance's group here in the University of Washington in the chemical engineering department. They're doing great work with these nanoparticles, but we're having trouble proving that they were actually having their surfactant, which is what they thought was affecting this process, sticking to the outside of the nanoparticle. So we were able to characterize that with TOF-SIMS, not something you can do with many other techniques like XPS, because PLGA and PEG have very similar atom percentages, and it would be difficult to pick out with ones that were just monitoring atoms or even bonding environments. But since SIMS actually can pick out different molecular structures, we were able to use SIMS to identify the PLGA, the PEG, and actually the presence of the P80 surfactant that they were using, and prove that they were actually having that surfactant associated with their nanoparticles as they were putting it into that environment. And so that was some nice work that we did with them. But we wanted to move on, not just for analyzing the samples themselves in spectroscopy, but more on an imaging. Can we use things like TOF-SIMS and XPS to really identify where are things, and not only where are things, but what is the chemistry of what is happening on, for example, biomaterials. So one of an old postdoc of mine, Mike Taylor, who has actually gone on to PNNL, he was working with polymer scaffolds and trying to see if we can use TOF-SIMS to give information about the chemical coatings that people are putting on scaffolds. And while I've shown so far TOF-SIMS of relatively flat surfaces of the DNA microarrays, and even the nanoparticles are just little nanoparticles that we put on a flat surface, a polymer scaffold can actually have a lot of three-dimensional structure in it, and it can be very difficult to analyze with surface analysis. So we wanted to know if we could say something about the 3D, because polymer scaffolds generally have a structure with them, like, for example, pores. They would have the cells growing in the pores, and eventually people were hoping that they could get like blood vessels to form within those pores. This is actually SEM of blood vessels that have been growing from an implanted scaffold from one of Buddy Ratner's groups, showing that they were actually vascularizing well. And so the size of the pores really matters, but also people might want to do a surface modification to change how whatever polymer they're using interacts with the environment around it. And so we want to be able to analyze that and help them know if they're making that modification. This is just a nice picture from Betty Ratner's group where they're demonstrating that their pores, in this case, they're having 40 micron size pores, have certain growth of cells in them and a certain encapsulation of the foreign body response that is different from the 100 micron size pores, which has a different type of foreign body response and a different type of collage encapsulation. So sort of their proof that the pore size matters, not just the material itself. And this is just a solid block of the polyhema that was used to make these samples. So even though they do these and they know the pores do matter, still there is a surface modification that you might want to have happen. For example, if you're making these out of materials that are hydrophobic and you want to do a surface chemistry to make your thing hydrophilic, or perhaps you want to do a bioconjugation chemistry, put a certain peptide on there, put a certain type of other sort of molecule onto there, that you might want to be able to say, yes, I created that sort of chemistry, not only from the outside of that porous scaffold, but all the way through to the center of that porous scaffold. The surface chemistry of the scaffold can affect things like protein absorption, the swelling of the polymer, and people do this in a variety of different ways, either just by chemistry or also by chemical vapor deposition or plasma vapor deposition. So we were working with people on that, and one thing we first found just by doing some basic porous samples is, here's an SEM of a porous scaffold. Just pulled this from a journal article, and here's one of our first TOF Sims images of a porous scaffold, and it looks somewhat like a porous scaffold, but TOF Sims is generally optimized to work on a flat surface. And when we're getting those ions from different levels of within the sample, different heights, it makes the signal very hard to understand. Plus it actually makes the spectra, and I didn't include that with here, it makes the spectra very broad. And so your peaks being broad, it's very hard to identify the exact position of those peaks and therefore the exact molecule or that it's standing for or that it's representing. So first we had to figure out a way to solve that problem by taking a scaffold and seeing what we can do to improve our TOF Sims image on those scaffolds. And so we had a scaffold. Initially, it looks like this when we take a TOF Sims image. It's very, very blurry. But by doing something called, by embedding it, very similar to what people do when they do embedding tissues for tissue slicing, for example, cryo tissue slicing, we would do that and use that same material to embed into the scaffold. We actually create a flatter surface that we can more easily analyze with TOF Sims. And so this type of, it's the same scaffold, but one is filled and one is not. Well, in this version, it becomes very blurry. In this version, we can see quite clearly where the scaffold material is in green here, and benzalkonium salt is just a salt that is an impurity within the embedding material, the polyvinyl alcohol glycerol material. So we're able to improve the image dramatically by doing our embedding process. So then we worked with some of our collaborators at Colorado State to help them identify if they were creating good conformal coating of their plasma deposition onto the type of plasma polymers that they were doing. So they had these polycaprolactone scaffolds. The pores were not as uniform as what is done in the Ratner lab. It's much more of like a spun mesh that is happening in here in very small pores. Then they would do a plasma chemical vapor deposition of these polycaprolactone scaffolds with a variety of different types of plasmas. If they wanted to have it hydrophilic, they would do one kind of plasma. If they wanted hydrophobic, they would do fluorocarbon, for example. So we studied the fluorocarbon version. So we wanted to see if we could identify how well they were embedding that fluorocarbon into the scaffold. We started by taking just a slice of the scaffold and doing some XPS at different areas around that slice. So there's the edge, the center, and the other edge to sort of quantify it with XPS, if possible, how much fluorine was deposited there. And then we did TOF Sims imaging to quantify or look at the distribution. And our XPS analysis showed that for the untreated scaffold, not surprisingly, happily, there is no fluorine. And we just see the carbon oxygen that one specs from polycaprolactone. And our high resolution carbon spectra shows also something similar of just hydrocarbon and carbon oxygen type carbon. 5 minutes of exposure to their fluorocarbon plasma, we start seeing a fluorine peak appear. And we start seeing fluorine peaks appear in the high resolution carbon data. And after 20 minutes, we see a quite strong fluorine peak appear. And again, stronger fluorine peaks appearing in the high res carbon data. So the question is this uniform across the entire sample? XPS indicated that by 20 minutes, it was fairly uniform across the entire sample just by looking at relative spots. And if you look at this top SIMS image, we have green here for the polycaprolactone, basically the monomer plus hydrogen. And so it indicates where our scaffold is, the polymer of the scaffold. And whereas red is a sum of fluorine-containing ion peaks that we see in top SIMS. And so, of course, for the untreated one, we don't see any fluorine, or I should say, happily, we don't see any fluorine. One never knows how that's going to work out. However, 5 minutes of treatment, we start seeing fluorine appear, and I suppose it's not terribly surprising, but we only see fluorine on the outer edges of the scaffold, and in the interior of the scaffold, we weren't seeing hardly any, if any, fluorine signal. And after 20 minutes, we were seeing intense fluorine on the edge of the scaffold and starting to see fluorine in the center of the scaffold. So the conclusion was, yes, they were finally starting to penetrate into the center of the scaffold after 20 minutes. However, there was some indication that perhaps there was a thicker layer of coating on the outside versus the inside by looking at the top SIMS images. So it's great that we can slice it and we can get that kind of information, but we wanted to take that a little bit further and actually see it in 3 dimensions. So we're seeing only where we hit the slice on that time, and it really doesn't tell us as much information as we'd like to have about conformal coating. And what we are doing here is utilizing the fact that top SIMS is not only for analysis, and an analysis beam is generally something like a bismuth 1 or a bismuth 3 liquid ion, a metal source, where that atom hits the surface, you lose some molecules off, and it embeds at a certain depth. But if you take a cluster of atoms together, in this case, this is a buckyball example, and you hit that into the surface, it actually makes a bigger crater, you reduce more material, and it doesn't really embed into the sample as much. And so you create this nice shallow crater. Sometimes that's good for analysis, although you can't necessarily always focus that beam as well. But it's very good for cleaning up a sample. So bismuth 3 is actually what we generally use for our analysis beam. So you can see it's sort of an in between the two. But what we'll do is utilize one beam, one type of beam for analysis, and another beam for actually doing a cleanup. So we'll have an analysis beam. In our case, it's probably bismuth 3+, but it could be a variety of different beams. You analyze the sample, and then you sputter and etch, and you remove a layer. So you'll keep going down in depth of analyzing, sputter, and then you'll analyze down. It's kind of like when you take cereal slices within a tissue sample. And so we'll create a series of images as we etch our way down that hopefully we can reconstruct to make a 3D image of what we saw in the sample. And this is sort of a pictorial example of that happening. Suppose you had a bead, or maybe it was a cell on a sample, a flat surface, and then you do a top SIMS image, and then you'll get a flat 2D projection of what you saw, even though there's topography there. And then you would sputter that sample, and everything would get a little bit smaller. That bead would get a little bit smaller. You take another image, you get another projection, and so on until, for example, you get completely rid of that little bead. And so you just get a little spot there at the end. So it sounds like it would work, but in reality, if you just match those up, reconstruct, put them all together, what you end up with is sort of a reverse image of what you actually had. So for example, if you knew spot blue was right at the substrate, when you reconstructed, or if you knew spot blue was right at the top, for example, when you reconstructed, it would look like it's at the flat of the substrate. And so you actually need to invert the image stack to get a correct profile, which is a little bit trickier than it sounds, but that's what we have to end up doing, is to invert the image stack to get the 3D to reconstruct correctly. And so we developed something called the Z corrector GUI, which will automatically do this for top SIMS data for those of us who do top SIMS analysis. And then we were able to reconstruct correctly 3D images from a variety of different biological samples. And this is just an example from a mouse muscle fiber that we did just to see that we could. And we were looking actually for some other different types of chemistry. But we were able to correctly reconstruct it and see things like the little cell nuclei. There's multiple cell nuclei apparently in a mouse muscle fiber, and then the fibrous region itself, and then the substrate. So by using that, we can also apply it to these porous scaffolds that we've been looking at. And so now, instead of just getting that flat 2D image of the porous scaffold slice, we can take a region and start doing that sputtering and analysis and sputtering and analysis, and at each layer, reconstruct that and get a more 3D image of how we're getting this conformal coating or fluorinated coating on a sample. And I find it's a little bit easier to look at this way. This is a flattened version of that 3D image. You can see the green represents the scaffold ion, an ion that represents the scaffold, and the red again representing where we see the fluorocarbon peaks. And the blue is representing the filler. Remember, we have our filler material in the background. And so in this particular image, I made the blue semi-transparent so that we can kind of more easily see the 3D image. Let's see if I can get it to spin. There we go. So if you can get it to spin, you can see where the red is sort of going up the sides of the scaffold. We can see it down here on the bottom. And so we're actually able to identify in more of a three-dimensional version where this coating is going onto the sample. So I think still some perfection needs to be done on this technique, but it's showing the possibility of what we can do with SINs to give chemical information in 3D, not just structural information, but actual information of where the chemistry is and what's going on there. So that works great for small distances, but SIMS has a very narrow range of where you can focus your beam. And if you start etching too far down, what you'll find is your image starts shifting. And so this is an example of that where we would, these are 20, oh wow, that's supposed to be micrometer beads. I forgot to change that. So it's not millimeter, it's micrometer. So 20 micron beads, and put them into a substrate, backfilled it with a polyvinyl alcohol. And so it's these polymethyl methacrylate and polyvinyl alcohol etching down there. And as we start etching down even 1 millimeter or 1 micron, there's a lateral shift of 1 micron. And when you start reconstructing those, if you didn't account for that lateral shift, what you'll see is what should be a round bead looks like a sort of oblong blob. So we worked on various ways to compensate for that. And one way you can do it is by actually using a GUI that we developed to shift the images accordingly, depending on depth. And then when you reconstruct, then your beads start looking more round, like an actual bead should. So that that's how we can accommodate for going further and further down. And if you go too far down, you have to actually accommodate by really raising the stage and positioning your sample. But with doing something like that, we can start accommodating for even larger and larger areas where we're doing our 3D analysis. So we've sort of gone with two paths. One is that 3D analysis with the polymers and for example polymer scaffolds or bioprinted polymers, but also looking at TOF-SIMS in relation to pure chemistry of the tissue itself. And a really nice set of experiments was done by my postdoc, Tina Angerer, who she also just recently left, unfortunately, off to a real job. Tina was working with a planarian flatworm. And as you can see, I like this particular image because it's this nice false colored image of TOF-SIMS key peaks that are showing all the different features of the worm. And this is a light microscopy image of the same slice of worm. So we can take a light microscopy image of the worm and then we put it in the TOF-SIMS and get TOF-SIMS image of the worm. And you can see all the different features coming out really nicely, even within just very couple cell, 2 cell thicknesses of these different areas within the worm. So we can sort of show the utility of TOF-SIMS for giving information about the chemical species that's present in tissues. One advantage is this is completely label free. So when people do normal histology, you have to do some sort of a stain, and you have to pick what you're going to target for. Are you going to target for a DAPI stain for the nucleus? Are you going to target you know, an H&E stain and get the collagen? And you don't have to do any sort of any kind of prep like that for TOF-SIMS analysis. And also there's the imaging technique of MALDI where you would have to apply a matrix, and TOF-SIMS doesn't require that at all either. So all you have to do is slice the tissue and put it on a substrate and analyze it. And with that, you get tons and tons of mass spectral data in imaging format that then the real challenge is actually analyzing it. We can see a lot of key biological species that people might be interested in. For example, lipids and fatty acids. We can see obviously any inorganics, which come out quite strongly in TOF-SIMS, and some of the small metabolites that people are looking for. So the work that we did on this worm was actually done on a slightly different TOF-SIMS. This is a J105 from Ion Optica. And whereas the other TOF-SIMS would bunch a beam, hit the surface, and then you get the analysis, the Ion Optica works a little bit different. It has a direct current beam hitting the surface. It takes everything coming out, and then it bunches them in the analyzer. And one advantage of the way this one runs is that you get very high mass resolution or mass, and also good mass accuracy, but very good high mass resolution. At the same time, you get spatial resolution. And for many of the other types of SIM systems, you have to, traditionally, you've had to choose between mass accuracy or lateral resolution. They are working on them to improve that, but this one has really exceptional combination of the two, and it does it fairly quickly, so we can get these very large images. We can also get imageable signals up to at least 2,000 Daltons, and that's for anybody who's doing biological work with lipids or proteins knows that the higher mass that you can get, the easier it is to interpret what that signal is coming from. So it works really well for things like lipids. And similar to the other mass spectrum TOF-SIMS, obviously you just focus your beam on different areas of the sample, and then each of those areas produces an individual mass spectrum. So we have that same result that we did last time. So Tina was focusing on the planarian worm. One reason we did this is just because they're very easy to work with. You can actually just cut a worm in half and you'll have another half a worm. We were actually going to do a regeneration study initially to see how the chemistry changes as the worm is regenerating this process, but found out that there really wasn't a lot out there on the chemical structure of the worms because most of the work was done so long ago that they were these really beautifully drawn diagrams from light microscopy and staining. So we wanted to start with just the whole worm just to give a background of what the worm should look like. And Tina was able to identify key lipid peaks within different organ systems of the worm, excuse me. And so this is what the mass spectrum would look like. And we've color-coded different peaks, not just the peak itself, but all of the peaks that are related to that particular molecule, for example, so that you can see kind of what we're looking at with our mass spectrum. So all this information comes out within the same mass spectrum, and we have to parse out which peaks are related to what, and then what those peaks mean. So we know that, for example, this peak related to all the other green peaks, and then we have to identify what is this green peak. So we do that in a series of ways. One is by imaging it and just seeing where is that peak showing up in the image. For example, 885 is showing up, and we're false coloring it here green, but it's showing up where in the drawn image one would see the quote nervous system of the worm. And by looking at the mass spectrum a little more carefully, we can relate it to a phosphoinositol peak with two different chained fatty acids off of that phosphoinositol. Similarly, we see different peaks related to focusing on where we would see what we might call the guts of the worm, the pharynx, and the reproductive systems of the worm, the oviducts and the testes, for example. So interestingly enough, we see different lipid species for all those. And then we can false color and sort of show where we're seeing all those peaks come out. But the big question that's always raised against TOF-SIMS is, that's great, but you generally only see fragments. Well, we've been seeing some of these intact lipid species. So that was great. But then the next question is, okay, so you see this intact lipid species, we see this mass, but the problem is this particular mass could be related to a variety of different lipids. If you're not familiar with lipids, they tend to have, there's some sort of head group, depending on the type of lipid or fatty acid it is. And then they have some sort of fatty acid tails. And they could be different lengths. They could have different saturations in them, a double bond. And that double bond could be at any point along this chain. And that could produce the same mass and yet still be designated as a different species. So how can we tell the difference? Traditionally in mass spec, what people do is they do an MS/MS, where they take this peak, they isolate it, and then they do MS on this peak. They break it apart to all of its different components to identify what makes up this peak. And we thought, well, SIMS just naturally does that because we hit an ion on the surface, it breaks everything apart. Perhaps we can utilize that. And so that's what Tina did here, was take her parent ion peak, map out where it shows on the sample, and then find all of the different images that also show intensities at that place, at the same type of image areas. And then find the different peaks related to that. So she started with, for example, the 718 parent ion peak and found a variety of different peaks, masses that had very similar images. And we worked with some friends at PNNL to help see if we could confirm what is going on here. So they did MALDI on a similar worm, looking for this 718 peak. And one advantage of MALDI is it does get higher resolution in the peak. And so they can take it down to 4 decimal places, whereas we were stuck with 3 decimal places. But it helps us verify that that is, yes, actually the parent, the ion mass of the parent. And then we did actual more traditional MS/MS of this, shooting a solution of the worm peaks through a traditional mass spec and doing mass spec analysis on that of GCMS, and being able to find all of those subsequent broken down peaks within more traditional MS/MS type analysis. And so by doing that, we were able to confirm that, yes, this is the species that we thought, and we were able to find it with more traditional mass spec methods as well. So we were happy to see that what we thought we were getting in TOF-SIMS was also confirmed by what we see in traditional mass spec. So ideally, at some point, we're working on being able to more automate this process, where we can just get an image from different ions, the different masses that we're seeing coming out of the SIMS, and sort of do an image matching to see how well they match and are they related, and then be able to put that together and say something about, is this going to be related? Can this be then considered a parent ion or one of the daughter ions of that particular parent ion? So in process of doing that, we've also been doing more just straightforward analysis of tissues. A lot of that has been related to cancer work that was done with a study done with people at the Fred Hutchinson Cancer Research Center. This was work that was just done on a mouse study looking at tumor progression and regression over time, and can we identify with TOF-SIMS some chemical information about tumor growth and regression. And so this is just showing that we did see some basic work of these tumors growing in, is a mouse pancreatic tumor growing over time, and then the presence of these blood lakes pooling we can see by heme in the chemistry of the TOF-SIMS analysis. So we were able to find a lot of chemistry within those tumors that are growing, and did a work with them also on some more breast cancer study of actual biopsies, where, and this is an example of a TOF-SIMS image, high resolution, and a serial slice of a traditional H&E stain, where you can see a lot of the same information. We're seeing bright regions where we see stromal. If we change up what we're looking at, we can start seeing identification of where we're seeing cell nuclei. But we wanted to be able to identify key components, so just the stromal region or just the cellular region of these tumors, to be able to compare them from patient to patient. And so we came up with a way to use PCA to separate out, for example, stromal region and cellular region. Why do we need to do this? This is an example of a tumor biopsy image, and we'll take TOF-SIMS at different parts of the biopsy. And whereas blue is where there's a high cellularized region, and you know there's a lot of tumor. For example, this is a solid tumor. Here's a tumor mass. but there's also diffuse tumor cells within here. And the amount of stromal versus the cellular cell nuclei part is very different in all these different regions. And so to compare from sample to sample, person to person, we wanted to be able to normalize that more by pulling out specific regions. And when we did that for our TOF-SIMS analysis of these are actual patients where we got their biopsy before they were treated with chemotherapy. And we just took the stromal regions from the biopsies of these patients and compared them with principal components analysis. We found that there's a certain amount of certain types of chemistry, for example, sphingomyelin and certain types of fatty acids that seemed to load more with the PCA analysis. Yes, I'm bringing back PCA analysis of the people who responded to chemotherapy treatment versus the people who did not respond to chemotherapy treatment. So we're hoping we're able to eventually lead into some understanding of chemistry of why some of these people, and this was a triple negative, actually, I don't think this was just triple negative. No, I think these were the triple negative patients, yeah, that responded to treatment, whereas some of them did not respond to treatment. And hopefully that could lead to some understanding of how they can improve treatment for people who didn't respond for traditional chemotherapy. So I will just summarize and finish off with, hopefully I convinced you that at Nessic Bio we are successful in taking some of these surface analysis techniques, turning them around with different protocols, data analysis methods, and modifying of some of the instruments to actually give useful information about biomedical research, not just for biomaterials, but actually in the tissues themselves. And also a little bit of a plug, because I am the director of the MAF, that most of the tools that we use are in the MAF and for people that to use on a for fee basis. And I actually have to acknowledge the MAF because they get funding as well. And so they helped with our funding. And acknowledgements for all the people that I worked with and some of my funding. And I would be happy to answer any questions.
Thank you so much, Professor Gamble. That was a really, really interesting talk. And we're open to questions now. I actually had a question sent to me from someone and would like me to ask that for them. They just might not be able to get on right now. So it's Dr. Catherine Granfield says, great lecture. What's the spatial resolution in the Z direction, depth, thickness during 3D profiling, etching, and how controllable and tunable is this?
That is an excellent question. I got to say, we are still trying to perfect that. I would say it's definitely not as good as we have in the XY direction. And so we do etch away a lot, but it's definitely submicron. I can assure you that. It's more on the nanometer range. But part of the problem with the way SIMS works is that it's really dependent on the material that you're etching through and how you set up the beam. So I can't put a real definite number to that. You could see within the, if I go back to, let's see if I can escape out of this. Oh, yes. If I go back to the one where we're looking at the little balls, for example, you can see the balls are 20 microns and we're seeing them with a pretty good resolution. So there's a lot of little pixels along here. So each little pixel was a Z as we're going down. So, because this is a side view that we're looking at here. So fairly good resolution for that, but not nearly the, like potential 100 nanometer resolution that one could see with the lateral resolution. I hope that answers. I know it's not a perfect answer, but I don't think there is a perfect answer for that one just yet.
Oh, that's great. Thanks. Thanks. Yeah, and I see Dr. Adranov has a question as well.
That was a great talk. Really enjoyed it. I had a question about sample preparation for your biological samples. So you showed the TOF-SIMS of the worm and the histology. These are beautiful images. And I'm just wondering what's involved in the preparation of the sample for the imaging, because I'm under the impression that you need to be under high vacuum. So do you have to embed the worm tissue or the breast cancer tissue in some way, or does it just go in?
Yeah, that's an excellent question. And I sometimes go into a lot of detail on that. Surprisingly, no, you don't. So if you've ever analyzed individual cells, you need to fix the cells with something. Usually people are doing like a glutaraldehyde fixation, or the cell structure will sort of fall apart and the guts will come out onto the substrate. It doesn't seem to necessarily be the case with tissues. And our theory is because there is so much of the structure around with the lipids and the connective tissue that it seems to support itself as it's in the vacuum. And we've seen this with proteins. If you embed a protein within a PEG layer, for example, the protein will stay in its 3D structure better. But if you just set a protein on a surface and it's not embedded in a PEG layer, then it will denature on a surface when it hits the vacuum. So possibly that's why I don't want to necessarily say that's going to always be true for all tissue samples, but within the resolution that we're looking at laterally with these tissue samples, we haven't had to do any fixation. That being said, the human tissues that I've done were always fixed in exactly the way that they would always do for cryo preserving tissues. That's because that was their procedure and they had to do it for a variety of other things. And so they do embed them in, they call it OCT. It's basically this compound that's polyvinyl alcohol and a glutaraldehyde or not glutaraldehyde, a PEG mixture. And they do that so they can freeze it because it's too small and they can't cut it and manipulate it unless they have it in a chunk of something, and then they can cut it. The worms were also embedded in OCT. I hate to say why, but because they're so delicate that there was no other way to kill, let's say, sacrifice the worm and keep it in its good structure and flat. So we actually froze them alive in OCT and then cut them. And that's horrible, but yes, that unfortunately was what we did. But in general, I have heard of other people doing SIMS where they just take the tissue itself frozen and then slice it if it's a solid enough tissue, and they can analyze it directly.
Okay, no, that's really interesting. Thanks very much.
I actually had a similar question and might just quickly follow up on say protein conformational studies. And I know you mentioned with PEG, you have better ability there, but what kind of steps can be taken if you are looking at something where you don't have that PEG surrounding it? And because I know you can get that protein conformational, really rich data from the top SIMs. And
yeah, if you don't have any, well, I mean, there's a lot of other ways that people have tried to do it, and putting like a sugar coating on, but not enough so you can just barely see it is one way. If those aren't your options, really you're stuck with doing a very light, like glutaraldehyde fixation of your protein. And I say really light because if you get too much of that cross-linking, it's going to affect your TOF SIMS peaks that come off. And so you'll see your amino acid peaks that you would expect from your protein will change as you start doing too much of that cross-linking, because you're getting too many different types of bonds going on in there.
Right, that makes sense. Thanks. I see another question from Mark Bissinger. Yeah, feel free to, yeah.
Hi, Laura. That was really good. I just had a question about the GUIs that you've written, the image shift and the Z corrector. Is that something that you've written that works with the IonTOF software, or is that something that you use in, say, MATLAB or something like that afterwards?
They're MATLAB-based GUIs. It's on our website. Actually, some of them are on our website and some might not be published yet. I'm not entirely sure. When they get polished enough, we put them on the website. Otherwise, they're in-house, but we can provide them for people if they want them. They are MATLAB-based right now. And so basically, anybody with TOF SIMS data needs to export it as a text file, and then they can import it into the MATLAB program and do whatever they want with the processing.
Are you doing your principal component analysis and multivariate analysis also in MATLAB? You're not using the onboard TOF SIMS?
Yeah, we've historically we've done our own. We've written our own sample because we already had that before they even had the onboard one, that we still tend to use our own versions.
Okay, thanks. Really good stuff.
Thanks.
I have another question about the nanoparticles. And so you've got the PLGA and then able to detect the surfactants on there, which obviously has been a big issue, being able to do that sort of thing. Is it possible to start to get any information on the density, say, of that layer around a nanoparticle or other materials?
Yes, it is theoretically possible to get that, but it is challenging. You have to develop your set of controls and analysis to come up with that. And I think that we're going to be working with the NANCE group on trying to expand and getting a little more information on that. But that is very challenging to get that kind of information, especially normally I would say, oh, go to XPS and we can get that information easily. But when it's all carbon, oxygen, hydrogen molecule, then it's a little more challenging to get that kind of information.
Yeah, definitely. That's still just getting the confirmation of it, of seeing the differences there even still.
Yeah, and we're hoping to get more orientation information as well. I think if we you know, like what is the surfactant sitting on the outside? Is it closer to the you know, core of the nanoparticle kind of thing? So a little bit of that information we're hoping to get more detail on at some point, but it's not straightforward analysis, let's say. Right.
I have another direct message, so I'm not sure if the person prefers to stay anonymous or not. I'll read it out, and if they want to add to anything or ask a follow-up, they're welcome to come on or send another message. So it says, I love the worm data. What is the variability between animals? Is this something you could use for phenotyping animals, for example, for animals with changes in lipid storage?
I sure hope so. Honestly, we've only looked at this one type of worm, at this particular planarian, the Jugusia, I won't even say it, well, anyway, so we've only ever looked at that one. And our next step was to actually cut off the heads and then watch the regeneration process, hoping that we'll see some changes, right? Chemical changes happen there. We have not yet looked at different worms themselves. If that was what that question meant. But I suppose it is theoretically possible if, you know, they have chemical differences. We're not looking at the genetics, we're looking at the production of the genetics, like what happens afterwards, like what lipids are made and what is formed where. So it's kind of like the second degree of that sort of thing.
Okay, they sent a follow-up and said C. elegans, please. So I think they're—
Oh, yes. All right. So
yeah.
So I would certainly hope that a C. elegans would look very different than this planarian worm. And I'd love to look at that. It's on my list of things that I'd love to look at, but we haven't actually looked at one yet. So I can't definitively say what it's going to look like.
Right. Really interesting stuff with the imaging here. Any other questions from the audience? Well, thanks so at this stage. Yeah, so if not, I think we will give another round of applause virtually here for our speaker. Thank you so much for joining us. It was really interesting and exciting to hear about all of that work and what's been going on with the toxins and XPS and the whole center there. And so
Thanks for inviting me. It was wonderful.
Thanks very much, Lara. That was great.